This method can help answer key questions in the genetic testing field, such as detection of mosaicism and liquid biopsy-based genetic testing. The main advantage of this technique is high-throughput detection of low-variant allele fraction. The implications of this technique extend towards diagnosis of tumors because detecting oncogenic mutations with high sensitivity can contribute to precision medicine.
Start this experiment by adding 10 microliters of DNA sample buffer to the tubes of an 8-tube strip. Then add 1 microliter of DNA ladder to the first tube of the 8-tube strip and 1 microliter of sample genomic DNA to each of the remaining tubes. Use microplate attachment to mix contents of the 8-tube strip by vortexing for 1 minute. Place the strip, pipette tips, and a gel device into an electrophoresis instrument. Press Start button to start the run. After the complete run, check the electropherogram on the display to confirm that the lower marker contained in the DNA sample buffer is correctly assigned on the electropherogram. If not, manually assign the marker in the electropherogram mode of the software. Then, in the region mode of the software, specify the size region of the genomic DNA to automatically calculate the concentration of DNA. Add previously designed primers,